But next, we're going to be having a talk from our keynote speaker, Dr. Linda Jo van Eldik. She is the director of the Sanders Brown Center on Aging and director of the Alzheimer's Disease Research Center at the University of Kentucky in Lexington, where I will be flying on Monday for a brain tumor thing completely unrelated to Alzheimer's. Her background includes a PhD in microbiology and immunology from Duke-UNC, which is exciting to have her be an alum, as well as a postdoc in virology and cell biology at Rockefeller in New York. She's received a number of honors and awards in her career, including notably a Zenith Award from the Alzheimer's Association, as well as an NIA Merit Award. She's a fellow of the American Academy for the Advancement of Sciences and a member of the National Advisory Council on Aging. Her research focuses on dysregulated neuroinflammation, looking for points of potential intervention to limit neurocognitive sequelae in patients with Alzheimer's disease. So she's going to talk to us today about her work and some lessons learned in Alzheimer's drug discovery. And so without further ado, I invite Dr. van Eldik. Thank you.
Well, thank you guys. I'll put this down where you guys can hear me. So I just want to say thank you for the invitation to be here. It's been really great so far, and I've enjoyed meeting all of you and renewing old friendships. And I just love this campus. I'm so glad you had the meeting here this year. I think that Pembroke is gorgeous, and everybody's been so welcoming and very organized meeting. I'm really impressed. So thank you for the invitation. All right, so if I can get this to work, let's see. All right, I think I've got it. So just a couple of disclosures. I'm listed as an inventor on patents that cover some of these drug candidates I'm going to talk about. I'm also the scientific founder of a startup out of Northwestern when I was there called Immunochem Therapeutics, but it's not relevant to the compound I'm going to talk about today. And also, I just unashamedly took some slides from Ted Spack from an ADDF conference a couple of years ago. All right, so what I'm going to tell you about today is some of our work on developing small molecule potential therapeutics that target the cytokine synaptic dysfunction axis of inflammation, and I'll explain what that is in a minute. And then I'm going to cover at the end some of the lessons that we've learned along the way. And I'm focusing it primarily for trainees and younger investigators, but it's probably relevant even to some of us old senior people too. So. Thank you. I may have to just say next slide. All right, so what exactly is the cytokine synaptic dysfunction axis? So normally in the brain, there are non-neuronal cells, the glial cells in particular, astrocytes and microglia. They're normally very neuroprotective. They surround our brains, take care of everything. But in many cases in disease or after injury, they become different states, an activated state primarily, where they can produce over-inflammatory responses, especially pro-inflammatory cytokine overproduction. And that can cause neuron and synaptic dysfunction, which then is an additional trigger for more glial activation. And so what we really need to do is try to develop orally active, brain penetrant molecules that are selective and that reduce these excessive inflammatory responses without being pan suppressors. We don't want to block the beneficial inflammation. We just want to block the disease or injury induced inflammation. Okay, so if you want to try to develop therapeutics, this is the classical sort of chevron type of thing that you see, where everything is in a linear row. Takes a very long time, 10 to 15 years it's been estimated to develop a small molecule from ground zero to FDA approval. And estimates are $1 to $2 billion per drug. And there's a high failure rate, especially in brain. So greater than 90% of all drugs that are being brought forward for CNS indications fail. And so our question was, could we try to reduce this risk of failure and enhance our chances of success, especially in academia where, we don't have $1 billion to spend on a campaign. Next slide. So this is kind of our drug discovery platform that we've developed, and I'll talk a little bit about each stage of this. But basically, you're starting with the design of your small molecule. And I'm going to focus today just on small molecule drugs, not on biologics, but the same principles will hold. So you need to design some kind of a library of compounds that you're going to screen for some particular properties. And I'll show you how we did that type of design. There are different ways to design and screen libraries. In the old days, people used to do very, very large, 200, 500,000 compound libraries and screen them for compounds of interest. We have a more focused approach to that, but you need some kind of a screen. And so there are different ways to do this. You can have a single molecular target that you want to develop a compound against, whether it's an activator or an inhibitor. Or you can do sort of functional phenotypic type of screens where you don't have a specific target, but you want to modulate some kind of a cellular process or biological system. In either case, you need to develop screens where you can screen compounds for their concentration-dependent and selective activity. And then those compounds go into the more complex stages. We've incorporated a pharmacology assessment stage where we're looking for compounds that actually have the potential to turn into drugs. So we're not just making compounds with chiral centers and very complex stereochemistry. We want to have simple compounds that can be easily made cost effectively. They need to be very metabolically and chemically stable. For us, we wanted them to be able to get into the brain, so we didn't even go forward with compounds unless they were brain penetrant. And then, of course, they need to be selective. They need to have potential for safety, so you don't want to develop a molecule that you're going to then put into an animal or a human and have all kinds of problems. So once that past that stage, then we go into the more complicated and more costly in vivo animal models. And at any point along this stage, you can, if a compound doesn't pass that criterion, you can go back and collaborate with your medicinal chemists and refine those compounds and then go through the recursive process again. Next. So I'm going to talk about just our single molecular target approach, where we were developing inhibitors of p38 MAP kinase, and I'll explain why we did that. But we also have a set of compounds that are non-p38 inhibitors, but they act the same way pharmacologically. They block injury or disease-induced pro-inflammatory cytokine overproduction. And so you might say, well, why did we decide for our single molecular target approach to target p38? All right, so when we started this, we thought, well, we're looking for a signaling pathway in the glial cells that will lead to pro-inflammatory cytokine overproduction in response to a stressor, a disease, or injury. And p38 is a very important stress kinase that's activated in response to different kinds of stressors, injuries, disease. And it had been known that it was a very important kinase that can drive biosynthesis of pro-inflammatory cytokines by both transcription and translation. And in this diagram, I'm outlining kind of a linear pathway where the p38 kinase is activated by a series of upstream phosphorylations, and then it is activated to phosphorylate its substrates, which then can stimulate the transcription of the cytokines. And for this talk, MK2 or MAPK2 is a very selective p38 substrate. So it's not phosphorylated by very many, if any, other kinases. And so we thought, well, this might be a good one. Now, this diagram is very much simplified, but you already see the punchline that in the next slide. This is really much more what it looks like. So this is really what p38 looks like. So signaling pathways, as you know, they're not all just linear by themselves. There's a bunch of crosstalk, lots of things interacting with other things. All right. Anyway, there's P38. So it's just sitting there amongst all the other signaling pathways. But we knew when we started the project, that P38 had been known to be important in both microglia and neurons. P38 alpha is the one that we're targeting. That's the isoform that is involved in the inflammatory control. And it's a kinase that's found in a lot of different cell types, not just in glia. But it had also been shown in neurons to be protective as well. So it's a target that could act in both microglia, astrocytes, as well as neurons to have kind of a dual effect by targeting a single molecule. But it hadn't been studied much in the brain. It has been found to be activated in multiple brain diseases, and especially early in Alzheimer's disease. And it had been shown that microglia that had P38 alpha knocked down had lower cytokine responses in response to different stressors, and that if you put neurons in co-culture with P38 deficient microglia, they were protected. When you stimulate microglia in culture with something like LPS and they produce the cytokines, those cytokines can kill neurons directly. And if you remove the P38 from those microglia, then the neurons don't die. And in peripheral tissues, P38 had been looked at by multiple pharma companies as a good target for peripheral inflammatory diseases like rheumatoid arthritis or Crohn's disease. And it was known that in clinical trials of other P38 inhibitors, there didn't seem to be, as a class, a problem with safety. If there were adverse events in some clinical trials, it seemed to be specific for the particular compound. So we thought, well, this might be a good one. The pharmas had developed P38 inhibitors that they didn't want to get into the brain because they were targeting peripheral diseases and didn't want to have any potential brain effects. So we thought, well, okay, we'll start with this. We'll start targeting P38. And so what I'm going to tell you about is how we got to this particular compound called MW150. What is shown on the left there is the kind of kinome tree. The little red dots are different P38 inhibitors that are out there on the market or, in different catalogs that you can buy or in different clinical trials. And you can see that these compounds hit multiple different kinases, not just P38 alpha. And when we started this and got our initial hit compounds here, it was already more selective than most of the P38 inhibitors that are out there. But it did hit more than just P38 alpha. P38 alpha, there's alpha, beta, gamma, delta, and P38 alpha is the important one that drives inflammation responses. So, but then as we refined the compounds for stability, for pharmacokinetic effects, for bioavailability, we got more and more selective. And then the clinical candidate, this is MW150, hits only P38 alpha. It doesn't even hit P38 beta. So it's the most highly specific P38 inhibitor that is out there. And actually, if you buy some of these P38 inhibitors, like these SB compounds, and to get out of catalogs, you got to be a little bit careful because they're not specific for P38. And so results you get with these compounds have to be interpreted carefully. Okay, so how do we start? So what we did was we started with you start with a core or a chemotype. It's like you pick what Legos you want to start with. So in our case, our chemotype is called a pyridazine. It's this ring with the 2 nitrogens on it. And we chose that because pyridazines can easily be modified at the different positions on the ring. And they can change the diversity and the pharmacology as you make reactions to diversify these compounds. So it's basically like you start with a core, you start with the Legos, and you kind of build it up, and you end up, with a big structure. that is then your set of compounds, and you can make lots of different compounds from that chemotype. We also picked that chemotype because many CNS active drugs had pyridazine rings in them. And so we thought, well, maybe that will enhance our chances of getting into the brain. So that's kind of how we started designing the small library. And we didn't have to use too many compounds actually to screen because we took advantage of a structure-based design. So the human p38 alpha kinase, the structure was known, and it turns out that that enzyme has a very unusual kind of a hydrophobic pocket that has a small, what's called the gatekeeper residue. It was a threonine, and it's very near the ATP site. And so you could design compounds that had very large rings. So here's the pyridazine, for example. Large rings like this phenyl, for example, that could fit into that hydrophobic pocket because the pocket was large because it had a small little threonine as the gatekeeper. And it was also known from other p38 inhibitors that you needed to make a nitrogen bond with the side chain of the kinase. And so we could take a pyridine ring here with a nitrogen that could make that side chain bond. And so we started out making those kinds of molecules, and we wanted to test this idea. This was our idea that, we put a nitrogen here, it would make a bond with the p38 side chain. We could have a bulky group here that could fit in the pocket, and that should give us good selectivity for p38 alpha. And so we tested that idea by making some analogs. And we would change the design. So for example, we could take away that ring altogether. And so then you wouldn't have that bond here. We could move that nitrogen down. So instead of here, we just move it down a little bit, or we could make that whole pyridine ring, we could move it up. And all of those should not be able to make that side chain bond, and they were all inactive when we made it. And then we also actually validated the design by doing a co-crystal structure. So we had the crystal structure of the p38, and we could make the compound and then co-crystallize them and show that the compound actually bound in just as we predicted. So the hydrophobic pocket and then the nitrogen side chain. And then the final sort of coup de grace was we could make a kinase, a mutation of the kinase, where we could change that threonine to a bigger methionine, which then should block the ability of this compound to bind to the kinase. Now, that single amino acid change doesn't affect the kinase activity. It's still a fully active kinase. It just should no longer be able to bind compound. And so we tested that by making the mutation. And we could show that in the wild type, of course, the inhibitor blocked just fine, the p38 alpha in a concentration dependent manner. But when we made that threonine to methionine, single amino acid change, then the compound lost activity. And these are different scales here. So it basically completely lost its ability to inhibit the kinase. So that was how we, made the initial set of inhibitors. We showed, for example, now we testing in cell culture, does the inhibitor actually block p38? So we had a microglial cell line, and we stimulated it with LPS, and then we showed by looking at phosphorylation of the substrate, MK2, that it blocked the phospho-MK2 in a concentration-dependent manner. It also blocked the ability of LPS to stimulate IL-1 beta in those microglia cells. And actually in vivo too, in a wild-type mouse, you can treat them with LPS, and we could show that it blocked the LPS-induced IL-1 beta production, which you see here. And that's well and good, but okay, so what about, oh, the cool thing, I forgot about this. So we had this mutant that had the T106M. And so this provided us with a potential way to say, okay, are we really hitting P38 in vivo? Because a lot of times, you'll take a compound and you put it in an animal or eventually in a human, and it may do something completely different than what you think it's doing. It may actually target something else. So what we did was we got a mouse. There was a mouse already that had a T106M mutation, and we obtained that mouse. It was from Merck. And what we did was then, and just as a reminder, once this moves, this kinase is fully active, but it can't bind compounds. And so we tested that in vivo. We took a wild type mouse, treated them with LPS, and we could show that the compound could block in a dose-dependent manner the IL-1 production in the brain of these mice, but in the mice that had the P38 alpha knock-in, it was a T106M knock-in, the compound lost the ability. So this showed that blocking of IL-1 beta in vivo was actually through P38 alpha, because if the P38 alpha could no longer bind the compound, it couldn't affect the IL-1. So there was a nice way of confirming that we're actually engaging the target we think we're engaging in an animal. Okay, but what about in real CNS diseases? Okay, LPS, that's great. That's an classical inflammatory stimulus. But, what about in animal models of Alzheimer's disease, which is what we're really interested in? And by the way, that mouse is reading Scientific American, so it's a smart mouse. And so what we did was we decided to look at the compound in different animal models that are relevant to Alzheimer's. So the particular one I'm starting with here first was an APP-PS1 knock-in mouse. So it was not a transgenic that overexpressed APP and PS1, but it had humanized APP and PS1 knocked in to the mouse APP site. So this was a prevention paradigm that we started with. And so we first characterized, does this mouse show an increase in cytokines? And we knew that it did. That's why we actually used it in the beginning, but we confirmed that, of course, you do see the normal age-dependent increase in cytokines that you see just in wild-type mice in this dotted line here. And the knock-in mouse had an even higher cytokine production in the red. And so what we did was we started the compound just as the cytokines were going up. So from several months of age, we treated them IP 3 times a week for 5 months with either the vehicle or the inhibitor. And then we asked what happens to the cytokines and the inflammatory responses. And we showed that the compound could block the IL-1 beta production, block IBA1, which is a marker of microglia, and 2 astrocyte activation markers. So it was blocking the inflammatory responses in these mice. And then we also used a different kind of a paradigm, more of a therapeutic paradigm. So we asked, well, could the compound work when the mouse already has pathology and the inflammation? So in this case, we used 11-month and 12-month-old mice where we knew the cytokines were already up, and we administered for just a couple of weeks, and then asked, okay, is it still able to block the inflammation? And yes, it was. It was able to block the upregulation of the IL-1 and a trend toward blocking the TNF-alpha. And in addition, it also blocked the cognitive deficits that you see in these mice. So we used a radial arm water maze test, which is basically looking at how many errors do they make trying to find a hidden platform. And what we found is that there is a deficit in these mice, and the mice treated with vehicle show that deficit, but the compound treated mice look much more like the wild type, so they don't make as many errors. So by blocking the cytokine production and the inflammatory responses, it had a beneficial effect on the cognitive deficits. So that was really nice. And then we thought, well, you know, what about other transgenic or other mice, Alzheimer's mice? Because what we wanted to do was, before we used a lot of money and a lot of time, we wanted to confirm in more than one animal model that the compound had an effect. So we collaborated with Ottavio Rancio at Columbia University, and he had an APPPS1 transgenic mouse. So now these are overexpressing the human proteins, and he uses a fear conditioning model, which looks at the ability of mice to respond to a tone and a shock, and they generally freeze when they hear it. The animal model has a deficit in this type of a behavioral paradigm, and the mice treated with the p38 inhibitor were much more like the normal mice. In addition, he also used the radial arm water maze, and you can see that the 150, the p38 addition looks much more like wild type. They don't show that deficit in learning. And these effects were dose dependent and very low doses. I believe we went down there to, I can't see it exactly, but maybe something like 0.1 mg per kg. So it's quite effective at low. This is quite a low dose for a mouse model. And the interesting thing was, in this model, there was no effect of the p38 inhibitor on A beta. In terms of A beta levels, we looked at both soluble and fibrillar A beta 1 to 40 and A beta 1 to 42. We looked at the plaque volume and number. This here looks like a trend toward a reduction, but it wasn't significant. But what we did see was we saw more microglia near the amyloid plaques. And you can kind of see this here in the double labeling, where you've got the amyloid plaque here in the red, and the IBA1 microglia are in green. And you can see there are more of them. It's like they're coming to the plaque, but for some reason they're not clearing it or not changing the amyloid itself. Different p38 inhibitors out there show differences on whether they or not they block amyloid, which I'm not exactly sure why. But then we thought, okay, well, what about a tau model instead of an AD model? And again, we collaborated with Ottavio. He had a tau model where he actually infused oligomeric tau into the mice, and then again looked at he's an expert in behavior, so he does all of these all the time, fear conditioning and radial arm. And again, the compound blocked when it blocked the lack of freezing, I guess you'd say, in the model, so it looked much more like the wild type. And again, they made fewer errors in the radial arm water maze. And this was just a small, a quick infusion. I mean, I was kind of surprised, but he took oligomeric tau and just infused it like 20 minutes before he did the testing. And I thought, well, how about like a real tau model? And so he also had a TG4510, a transgenic model, and it's the same thing. The p38 inhibitor had the beneficial effect in the fear conditioning and also in the radial arm water maze. And this was more like that's sort of typical dosing where it was for 45 days. And sort of just to like the icing on the cake here is we collaborated with another group, Bruce Lamb and Kieran Bhaskar, when they were at the same place. That now they're both at different places and both independent people, but they had the H-tau mouse model, which is a human tau transgene on a mouse knockout background. And they also did a therapeutic type of paradigm where they used quite old mice, 20-month-old mice, and administered for 14 days, either the vehicle or the inhibitor. And in this case, so they looked at target engagement. They showed that the compound would block phosphorylation of its substrate, MK2. It blocked the production of IL-1 beta and TNF alpha, which you also see in this model. And interestingly, in this model, you do see an effect on that pathology, on tau pathology. So it blocked both tau phosphorylation as well as tau aggregation, and it had a beneficial downstream effect where it prevented the loss of synaptophysin, which is a synaptic protein, and it prevented the Y-maze behavioral deficit. So again, by blocking the p38 inhibitor, you have all of these beneficial effects downstream, both on the inflammatory response as well as the neuronal synaptic responses and functional effects. So then the 150 has actually gone further on. A lot of selectivity experiments have been done. It is a completely specific kinase targeted compound, at least in all of the kinome-wide screens that are out there. It only hits p38 alpha. It doesn't hit any GPCRs that are in these typical screening panels. It's not an agonist or an antagonist of more than 166 GPCRs, and it doesn't hit any of the historical targets that many compounds will hit off target, like ion channels or transporters. They also confirmed that it is orally bioavailable. It's brain penetrant. It has linear exposure and kinetics. In a single ascending dose study in humans, it has appropriate PK. So it looks like a great compound, and we're moving forward with it. And the next steps, of course, for Alzheimer's disease, it's really complex because it's difficult to know, and there's not really consensus in the field, exactly when is it best to target inflammation in Alzheimer's disease or any dementias, any chronic long-term disease, for example. What we need to do is we need to figure that out. How do we select the right subjects to go into an anti-inflammatory trial? Are we going to need combination therapies? Since that is a complex disease, I sort of think of it like HIV, when you had one drug that targeted the kinase, one aspect of the virus, it wasn't very effective. But when you got the cocktails that hit different parts of the virus, then it was effective. It's probably going to be the same way in Alzheimer's. Since it is a complex disease, we may have to have an anti-amyloid, maybe an anti-tau, an anti-inflammatory, maybe a mitochondrial or metabolically targeted compound. So I think we're a little bit early yet in that field. We need to figure out the best therapeutic window. Should we administer it early? Should we administer it later? What combinations? And then how do we choose those people to go into the clinical studies to potentially enhance our chances of seeing an effect? But this compound is moving forward. It's being developed for an Alzheimer therapy. It's been in the long-term preclinical tox in the 6-month rat and 9-month dog studies with no issues. And so it's moving forward now into an early MCI-AD trial. So I wanted to just talk a little bit about what we learned along the way, because drug discovery is difficult. We've been kind of lucky. We've had 3 compounds. We've had this one MIMW150, which is the p38 inhibitor, but we also have 2 other compounds that don't hit p38, but they work pharmacologically. They work the same way. They block the biosynthesis of cytokines in response to injury or disease. They just act through different signaling pathways. So I showed you all those converging and diverging pathways. Those 2 compounds block other signaling pathways that lead to cytokines. But by blocking biosynthesis, we have long-term downstream effects because it's a pharmacodynamic effect. If you block something upstream that causes something downstream, then the downstream stuff doesn't have to happen, and your compound doesn't even have to be there anymore. So those 2 compounds, one's in an intracerebral hemorrhage trial, and one's in a trial of patients with brain metastases who get radiation therapy. Both of those kinds of injury cause inflammation and cognitive effects. So we're testing in those more acute disorders where we know the therapeutic window. We know when the cytokine surge happens and when to target it. So what did we learn? I'm going to talk about these three sort of major lessons for drug discovery. So first of all, you need to balance your risks. You've got to realize that not just in drug discovery, but in almost any research project that we're going through, we're not going to have success with every experiment. And in fact, probably more things will fail than will succeed, but you're going to learn something along the way with every failure. So what we like to say in drug discovery is to fail fast and early. Figure out, make the best decisions you can to optimize your variables. We integrated sort of smart chemistry and smart biology so that we made drug-like compounds from the very beginning. But figure out what it is. Where are you going? For drug discovery, what are you going to use as your translatable biomarkers or your endpoints? What are they going to be, and are they going to reflect the mechanism that you're targeting? So that's really important. I love the way it works sometimes and not others. The next important thing is build the right team of people. You really need interdisciplinary teams that have multiple perspectives, multiple roles. I really like this kind of cartoon where everybody is looking at the elephant from different perspectives and says, and so these men of Indostan disputed loud and long, each in his own opinion, exceedingly stiff and strong, though each was partly in the right and all were in the wrong. So, you can come into a team and you have your expertise, you have your opinion, you have your thoughts, but if you don't work together, then you all could be wrong and you all don't go forward at all. It also takes a lot of people, okay? It takes a village to build a team. Now, of course, you're going to have a PI or, a CEO if you're in a company. So that's the godfather. It's for all of you PIs out there. You're the boss, the buck stops with you. You have to make the final decision. But you've got a team of people that have different expertises. And so, of course, you're going to have, Ann Landers for human resources. You're going to have important Scrooge for finance. You're going to have Perry Mason on, figuring out if you're doing everything correctly and legally. You're going to have, the Pink Panther here for regulatory. You're going to have Clint Eastwood for your development team. And of course, Gene Wilder is important for that wild and crazy research idea that you're going to have. But to be a little bit serious here, I don't know how many of you people know who this person is, but this is Solomon Carter Fuller. And he was an African-American physician who worked with Alois Alzheimer and was working with Alzheimer on his original team. And so, Alzheimer had a super team with diverse expertise, different perspectives. Of course, they were all male in those days, but, at least he had a group of people that we only hear about Alzheimer's and think about Alzheimer's himself, but he had an interdisciplinary team that worked with him. So that's really important. Okay, then you got to figure out, how are you going to do the project? I mean, how are you going to get the funding to do this? We can't all just go out and beg on the street, but, you think about different sources. Of course, if you're in academia and you can get grants or fellowships, talk with the people who are relevant, talk with your program officers at NIH, think about all different kinds of opportunities. Get feedback on your ideas, make sure you get people to read your grant proposal that you're going to put out, and get advice from people that you trust, because it's really important that your friends give you the critiques ahead of time before you spend 8 months waiting for reviews from NIH and then have to turn around and send it all back again. And then, of course, weigh the benefits with any potential restrictions that you might have. So there are some organizations that may give you money, but may require a lot of stuff in return. So think about the cost-benefit ratios as well. And then the most important takeaways on all of the lessons learned, I think the team is essential. I mean, it's crucial. I could not have done any of this work without our wonderful, medicinal chemistry and pharmacology colleagues, as well as all of the people in my own group, all of the collaborators who had expertise in different aspects of the project. You need to communicate very clearly. These projects are expensive and animal models are expensive. That's more my expertise, but the human trials are unbelievably expensive and complicated. You've got to be flexible and you got to be able to collaborate. And again, fail fast and early. If something's not working, don't beat your head against the wall all the time. Maybe come around the other side of it and see, you don't have to go through the wall, go around the wall. But sometimes you just have to fail and start something else and really plan ahead. It's amazing how much, how long everything takes. It takes a lot longer than you think it would take for anything, it seems like, in science. Stay focused on whatever your goals are. I mean, science is great. Science is fun. It's, you can run the risk of getting distracted and follow, something that's look real cool and interesting, and then forget that, you got to get that PhD and, in a few years. So. All right, so talking about our team, we had wonderful groups of people. I've mentioned some of them here already. I didn't mention any of the clinical folks, but we have a great group of clinical trialists and clinicians. And actually, Danny Laskowitz here, is that Duke? So if y'all see him, he's collaborating with us, and our funding agencies, without which we couldn't have done any of it. And most importantly, we have postdoc positions available at University of Kentucky. We have a T32 called Triad, Translational Research in Alzheimer's and Related Dementias, and we have predoctoral slots and postdoctoral slots. And I would love to have any of you guys who are finishing up and thinking about a first or second postdoc, contact me and let's talk. So thank you very much.
Yeah, great talk. Thank you. You made the point early on that inflammation is sort of a double-edged sword. You need it under certain conditions, particularly under conditions of an infection. Have you guys tried looking at the effect of this P38 inhibitor on animal models of infections?
Well, we had the LPS model. That's about as close. We haven't done any sort of real infectious disease models. I should say that in the other project, the non-P38 inhibitors, but that work the same way, in one of the human trials, so we've done several human trials with these non-P38 inhibitors. They did an endotoxin study in humans, which is sort of interesting. Low dose LPS given to humans induces kind of a sickness behavior, almost like the flu. And they collected plasma over the first 12 hours after LPS and measured cytokines. And it was only 8 vehicle and 8 drug. It was double blind. And I thought, well, nothing's going to change with just 8 people in each group. But they measured 6 different cytokines. 4 of them didn't change. There was an effect of LPS, but there was no change in the level of the cytokines. But TNF alpha, which is pro-inflammatory, went down, and IL-10, which is anti-inflammatory, went up. And I thought, well, that's kind of interesting. So, maybe it's actually, you're able to see changes in plasma as well as in brain or CSF. So we're kind of moving forward thinking that maybe we can use plasma cytokines as a sort of a translatable endpoint.
Thanks for the talk. My question is a bit naive. So I'm wondering, like, these inflammatory response, usually they have a role. So at the question, when is the right time to give these as a therapeutic? How can you understand? Because I'm wondering now, also with the COVID situation, what is the mechanism behind, can these drugs being taken at the wrong time and have a detrimental effect instead?
Well, that's why we try to develop drugs that have a high potential for safety. Now, what you're talking about is really not off-target effects, but on-target effects, but at the wrong time. And that's why I was saying it's really hard in chronic diseases, chronic inflammatory, or diseases that have a chronic course and inflammatory responses at different times. It is difficult to know. I mean, until we get one that actually works, I think it's going to be more effective early in disease. And there's a lot of reparative stuff that's going to be going on later. But in Alzheimer's, it's hard to know. That's why we are focusing on these acute disorders, because in hemorrhagic stroke or in traumatic brain injury or in radiation therapy, you know when that cytokine surge happens, and you know that that's the bad guy. I mean, we know that from animal models, from other things. And if that's a single insult, like the stroke or like the TBI, if you block that cytokine surge, you can stop the drug and you can still have long-lasting cognitive benefits. We showed that in an animal model of radiation therapy, that you can administer the drug while the radiation treatment is going on, but then you can stop, and 60, 90 days later, you still have cognitive benefits because you don't have the inflammation continuing. You just had that inflammatory response. But in a chronic disease, especially in elderly, they've already got a higher level of inflammation just at basal. And so, how do you find the right people? It's complicated, that's why we're thinking going more toward the personalized medicine idea, that maybe we'll have to pre-screen people and get people with a certain level of inflammation already, and then bring that down, and that those might be the individuals that you would have the most benefit. Or ideally, I think our drugs are going to be more effective more as a preventative or delaying onset, by controlling that just sort of normal age-associated increase in the chronic low level of inflammation that's higher than normal. If you can control that, kind of like a statin or a low-dose aspirin, maybe it's, maybe when you turn 40, you start taking our drug, and daily dose of low dose of our drug, and then you don't get dementia later. But how you could never really show that. That's something that you couldn't do that before a drug is actually approved. That'd be way too expensive and time consuming.
Oh, I may have missed this, but I think at the beginning of your talk, you said that P38 alpha was active in both neurons and microglia. Is there a readout you could use to see if the neuronal P38 alpha activity is also inhibited and potentially contributing in some way to the cognitive benefits that you're seeing?
So we haven't done that in animals. You could do a cell-specific knockout in P38. We didn't do that with the neurons. We only did that with the microglia monocytes. But in culture, we did primary neuron cultures without any glia anywhere, and just used three different toxic stimuli, neurotoxic. So one was excitotoxic stimulus, one was a sodium nitroprusside, like a NO type of thing, and one was oxygen glucose deprivation. And when we added the P38 inhibitor to those neuron cultures, it blocked the toxicity of all three of those neurotoxic stimuli. But we haven't done anything with animals to do that, but just in neurons alone, in cells.
Yeah, I guess my question is a little bit related to that. Is you could imagine just whole animal inhibition of P38 could have detrimental side effects in some concept in some way, whether it's in neurons or other cell types. Do you have any evidence that acute versus chronic delivery might show opposing phenotypes?
I mean, not really. I mean, in the animal models that we've done both the long-term one, the 5-month administration, as well as more acute administrations, and we didn't see any obvious detrimental effects on the animal in terms of health or other sort of endpoints. But we haven't really, looked specifically in the pharmaceutical kind of experiments that companies have done with other compounds. There didn't seem to be any class effects of blocking P38 alpha. And there is a P38 alpha inhibitor in clinical trials right now for Alzheimer's disease. It's a small company that's developing a different inhibitor. So I don't know. I think it's all about the therapeutic window, and pharmacology is not just like how much drug you give, but when you give it and where it's working. And so I think it's going to be dependent on the disease indication as well as your administration paradigm. And so it may be that P38 inhibitors work better for certain kinds of indications and not others. And P38 may be really important in some diseases where it's beneficial and you don't want to inhibit it. But that's the nice thing about a small molecule. You can start and stop therapeutic administration. So if you need to do sort of staggered administration paradigms in a clinical study, you can also do that if it turns out you have to do
Has
that.
anyone made P38 alpha knockout mice and done a similar assessment in an AD model?
So in the P38 alpha knockout, generalized knockout is actually lethal because P38 is really important in development. And so you can't just knock it out globally. And that's why we had to use cell-specific knockouts or knock-ins of certain, in certain time frames. But we've done a P38 beta global knockout. It doesn't do anything. It's just, it's alpha that's the key kinase. But there's not a whole lot still known about exactly what does P38 alpha do, just, when it's normally, it's just its kinase, kind of basally. And when it gets activated by other activating stimulus, is it important only as an inflammatory driver, or are there other P38 functions that, we don't want to block?
I was wondering if you could talk just a little bit about your experience with sort of blood-brain barrier permeability, which is always an issue. And it seems like, in some of these scenarios, a lot of it starts with intelligent medicinal chemistry design, right? But then, at some point, you have to put it in something and see if it gets to the brain. And, maybe that in a mouse tells you something about it in humans. But then, what in the human context are you sort of doing? Are you looking at it in CSF? Are you, we do stuff with brain tumors where we can cheat because we give it pre-surgically and then we just look in the tumor because we're going to take something out, but that doesn't work necessarily in the sort of dementia space. And so I was just curious.
Well, in the animal models, we actually did do in mouse, we did brain to blood ratios and all that. In humans, we are in these human studies, we are looking, for example, in the ICH, we are taking CSF if the patient has an intraventricular catheter and getting CSF collected anyway, then we'll be able to do the PK in both the plasma and the CSF. But, it's difficult in a phase 1 or phase 2 trial. You can maybe get CSF in some of the trials, some of the Alzheimer trials, it's fine to get CSF. But in the ones we're doing, like the radiation therapy trial, we're not taking CSF. Those patients don't get CSF collected. But in the stroke trial, we are when we can. And then we'll be able to directly show that the exposure in CSF is there.
Hi, Linda. Thanks for coming to Pembroke and giving some great chemistry and drug discovery for everyone to experience the chemistry. And this is beyond my bandwidth, but knowing about inflammation and the teams at Gladstone Institute, that many strokes and seizure events will drive Alzheimer's and dementias, which may go up and down at times. So in the future, is there ideas of continue monitoring somebody where have a monitor on me that tells me what inflammatory signals are, and my drug application is based on what I'm having? Because the timing question was very interesting about inflammation.
No, I sort of like that idea. I think that the nice thing about being able to take CSF or plasma and do fluid biomarkers is that you can get these profiles. You don't have to measure just one thing like you do with, let's say, a PET scan or, an MRI, imaging sort of biomarkers. You can see a profile of inflammation, and you can kind of look at the anti-inflammatory, the pro-inflammatory, sort of the good guys, bad guys, and see if you can develop a signature that is predictive of either progression or, diagnostics. So in our Alzheimer's centers, we're focused a lot on fluid biomarkers to do that very thing. And we've looked at our cohort and we can, put people into tertiles or quartiles in terms of their profiles, both vascular profiles as well as inflammatory profiles. And that's kind of where the field is going, is, in terms of this sort of more personalized approach rather than lumping everybody into just based on their cognitive profile.
Really good discussion. But I want to add one last thing, and that is when you turn on an inflammatory system, metabolism in the brain goes haywire.
Oh, yeah.
And so a combination of drug and figuring out how the metabolism of the brain has changed might make it easy to decrease the pharmaceutical impact of the drug, at the same time being able to boost the metabolism of the brain.
Oh, yeah.
You can see it below.
The immunometabolism is a big area right now.
And it's really important.
Yep.
Thank you.